Good afternoon. This is day 4 of Y 2025. We are here for the Reverse Engineering Live Afterparty. I think most of you already know what's going to happen. If you have a question, if you want to talk, please raise your hand, your feet, your tentacle, whatever. Make yourself known. I will get a microphone to you so we get the question or the comment on the recording and on the stream. And otherwise, I will hand it off to Bert. And please welcome him with a very loud applause.
So, thank you so much for showing up. This is a bit of a weird presentation that was born in 2017. And then I did the first presentation on DNA here, and I had like 120 slides. Because I wanted to tell everyone everything, and it didn't fit. So then we sort of, as an emergency procedure, said, well, we're going to have a second talk where we're just going to do the rest, and there is time for as many questions as you want, and you can ask me about your own theories. And in the slides from yesterday and the day before that, I skipped a few slides, and as a sort of teaser, I'll start talking about those. But if there are no questions after that, then we'll done in 10 minutes. Which is nice, because then we can all move around and get into the fresh air. But as long as people have questions, we can just go on and on and on. And it is really, this session is really meant for people with questions. And yesterday we spent almost an hour after the talk doing a sort of after party next to the tent. So maybe there are some questions, but as a teaser, I'm going to start with one of the slides that I skipped over. Unless there are already some questions. Yes?
So, in the talk yesterday, you were talking about this...
Can you be a bit louder, because we can...
Yeah, in the talk yesterday, you were talking about the correlation between the numbers of the different bases in a string of DNA, like A and T, C and G.
Yeah.
And that nobody knows why.
Yeah.
But is that not just because the other one is the complement of it? And
yeah.
Why is it not just that?
So I'll repeat the question. So one of the things that I reported yesterday is that if you have a genome that for some reason the number of letters C in there is almost equal to the number of letters G in there. And if you look at the way DNA works, that you could sort of expect that. So I have the right... Yeah, here's the one. So DNA is like a big tape of life. But different than normal tape that we know, you can read it from both sides. So you can read DNA from one side in this direction and on the other side in that direction. It's not like the tape that we use for our backups. But it is a redundancy mechanism. So if there's an A on one side of the tape, there will always be a T on the other side of the tape. And if there is a C on one side of the tape, there will also always be a G on the other side. This is used for redundancy. So if the DNA gets damaged for some reason, then there's a missing letter, and then life looks at the other side of DNA and finds the backup there. So if there's a missing letter, there is a G on the other side, it knows I should put a C in here. In this way, it is of course immediately true that if you take a two-sided string of DNA, there will be absolutely identical numbers of A on one side and T on the other side. This is a mathematical principle. So as long as the DNA is whole and valid, this will be true. But the weird thing is now that if you split up the DNA, and you say, okay, now I have single-stranded DNA, so there is no other side, then it is still very true within 1%. So we are not only finding that there is the same amount as A and T in DNA in total, but also on each side individually. And that is almost always the case in life, with a few exceptions where it does not hold, but it's almost always true. And really no one knows why. So we know why it's true on both sides, but we don't know why it is true on one side. And there are people that say, well, it's trivial, that it should be like that, but these people never follow up their statement with an article in which they exactly describe why it is trivial that it's like this, and they especially never also describe why it is true for longer strings. So what we have found is that if you have the CTA in DNA and you look at what would have been on the other side of the DNA, these frequencies are also completely identical. So this is like when you would scan a binary, in a computer binary of a computer program, and you would find that a certain opcode of 3 bytes happens for a certain amount of time, and then by some reason of magic, the reverse of that opcode happens the same amount of time. And this is still an open thing, and it actually not only works for combinations of 3 letters of DNA, it works, as far as we know, for up to 10. And I'm still on the fence if the answer is maybe, yeah, this is just how random works. But it seems weird. And if you, it's not just me. If you look at the Wikipedia page on this, on the SharGov's rule, it also says, yeah, we don't really know why this is. And, enjoy. But yeah, that's a very good question. Yeah.
Yeah, so perhaps this is something to do with the structural integrity of the DNA to retain it.
Yeah.
Because it also folds up in 3D at some point. It's not a very long string just floating there.
Yeah, so let me this could well be true. And I need to show you this table again. When you take 3 letters of DNA, it corresponds to an amino acid. And these are the building blocks of life. So you have this tape of DNA which has instructions for the 1D ribosome printer, 3 letters encode for a single amino acid. But it turns out that since we have these 3 letters of DNA, we have 4 possibilities times 4 possibilities times 4 possibilities. So we have 64 possible codons that encode for only 20 amino acids. That means that we most of the time can pick between 3 or 4 codons for the same amino acid. That means that nature has some flexibility. So let's say there is a charge, let's say the DNA molecule would become too positive if there were too many C's in there. Then there is a force that says, okay, well, if you ever mutate this C into a T, but it still means the same amino acid, then that would be favorable. So there is a room for nature to be a bit poetic, or speak a different dialect, maybe so that the charges don't build up. Or there are other things that are like really bad news. We know that DNA, the A feels attracted to T, the C feels attracted to G. Let's say you would have a piece of DNA that says C, 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 and then G, 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 G. You can imagine that piece of DNA wants to fold up. That would be very bad news. And this variation in encoding allows nature to sort of escape such traps. Now, this may sound like a very biological thing, but if you remember the old spinning Rust hard drives, so not the programming language, but the hard drive. If you would have a string of consecutive ones that was too long, then the drive started to become a little bit unsure, have I just seen 5 ones or have I just seen 6 ones? So they too had to massage the data a bit, so it would not become one giant North Pole.
We do the same in radio. It's data whitening in context, yeah.
There is actually a lot of tricks that we have discovered with our puny human technology and that have been in use in life for the past 2 billion years already. And the cool thing why we should look in this so much harder is we have now uncovered parts of our own technology in DNA already. Like the virus scanner I mentioned yesterday. If we had known about that before, it would have been very inspirational. And I'm sure that there are further secrets or techniques hiding in there. So, for example, we know that when we have a control theory, if you have a ship or an airplane or whatever, and you want to keep it within certain limits, we know that you should not just move the steering wheel around because it will start oscillating and life will become very unhappy. And at one point in the 1930s, we discovered this theory ourselves. as puny human beings. And then later we found out that nature knew about this for the past billion years already. Because, for example, the pH, the acidity level of your blood, is managed now that we now know with a 5th degree polynomial. And which is like extremely luxurious. And nature has been doing that for like 100 million years already. And we are only discovering it now. So that's some cool stuff. Yeah.
Hey, I would like to give a possible answer to one of the previous questions. And if I'm wrong, I would love to hear why I'm wrong.
And also, just to interrupt a little bit,
yes.
I also do not know everything. So please do not misunderstand. I have read a lot of stuff, but please know that I'm aware of my limitations here. But I hope you come up with something great.
Okay, hopefully, but it's really simple. I think. Hopefully, I'm not one of the biologists that says that and is wrong. So about the abundance of AT versus GC. So G and C is because of the thing that the base pairing. Yeah, exactly. So nature would want as many G's as C's and as many A's as T's because you always need the two of them.
Yeah.
So these are like linked in abundance to each other, I'd say. And then there's different reasons why nature would want different ratios. So for example, G and C makes DNA melt at higher temperatures because it's linked in the DNA strand closer together, and you want DNA melting for different purposes. So if you want to read it, you have to split it apart, you have to melt it basically. So there's reasons why you would have varying contents of the 4 bases, but A's and T's should always be in the same concentrations, and G's and C's. But now, like, nature has those different ways with the triplets of like saying the same thing, and so it's kind of random. Like, when do you have A's and when do you have T's, for example? But still, the ratio is linked. So I guess.
I think so. You have sort of summarized the state of the biology insight by now. So what you said is what many people say. They say there are reasons why it could be like this, and why it could even be useful. But also, and I'm not making fun of you because you have just reached the equivalence of the whole biological insight so far. So, but if you, in your description, you already said, yeah, nature should want this, and this would be nice. And for now, this subject lacks a certain rigidity. And I present this graph also on the right. Hope that you can see it. You can see that the deviation between C and G is between minus 0.5% and plus 0.5%. This is quite tight. So we need sort of arguments that are a bit better than well, it should be nicer than that, because it does keep it within 1%. Now, so the thing is, and I invite you then, okay, and I've been telling this to many people. So, okay, if people are sure of this, write a paper. And write a good one, a solid one, in which we run the simulations. For example, it would be lots of fun to first start out with completely random DNA, and then and people have simulated, for example, this. You start out with completely random DNA, and you posit that every once in a while a bit of DNA gets copied in the wrong direction. It turns out if you do posit that, you end up with this scenario. That could be enough to explain it. More interesting, of course, are also the deviations from this rule. Because like you said, the G and C, sometimes you want to have, if you have a lot of G and C's next to each other, you have very tightly bound DNA, and it will not, it's tricky for nature to tear it apart. And if you have a lot of AT, it's a lot easier for nature to do that. So you find that in some stretches of DNA, for example, where a gene begins, there's a lot of A's and T's because it makes it easier for life to tear open the DNA and convert it into RNA. The deviations are quite strong. And now to make it even worse, of course, you can have these bulk statements why C and G need to be sort of similar patterns. That still does not explain why the longer strings are also so closely bound. If you head to the Wikipedia page on this, which is linked from the slides that I'll show the QR code later, they even mention a third rule, the Sierpinski rule or something, which is another sort of thing where the letters adhere to a certain pattern and that no one knows why. So there is room for if you're good with bits and bytes, which I think we are, you can have a lot of fun here. Yeah. Yeah. Oh, there's also one.
You want to follow up? Is there? Wait for it, if you don't mind. You want it on the recording?
Yeah.
To the mouth, please.
I wondered, is there data or the thing that we can download from your website? Is there data to compare these sequences in the age of the bacteria or the temperature where they live and see if there is a correlation between the appearance of these sequences, if they are the same? Because because of the suggestion, maybe first they are just copied.
Yeah. So I'll repeat the question because I'm not sure if it's made to the recording. Is there a way to correlate the data from skewdb.org, which is my site where you can download a ton of CSV files with all kinds of data about bacteria? Can you correlate these patterns to things you know about the bacterium? To make that possible, so yes is the answer. In the CSV file, we assigned a number to every species known to man or to woman, and that's a taxonomy ID. And if you then know the right FTP server, you can link that taxonomy ID to the bacterial family and all kinds of other things that we know about that one. And my scripts also download that taxonomy database, which, as is usual for biologists, is in a terrible and hard to parse format. I'm not sure if these people were inspired by DNA. They say, well, DNA is hard to read, so will my database be. But I don't know. We need to introduce these people to, I don't want to get them hooked on JSON. Actually, so I'm not telling them about that, but if they could just ship their stuff as a CSV file, maybe it would be very nice. So, but the answer to that is yes, and I've actually done some of that stuff. Where you find that, okay, biologists are, if we compare to the open source community, who always rename things and start new libraries and stuff, every few years the biology people go like, let's rearrange life. And so at first there were bacteria called firmisuits, and at one point they said no, that never happened, and they invented completely new family names for the bacteria, which fucked up all my scripts. This is a trend in biology, by the way. So if you just have your scripts and they work, and you go away for 6 months and you come back and everything is different, and you don't know why. But you can make these comparisons where you say, I want to see how all the firmisuits or the bacteria formerly known as firmisuits, how they behave. And then you find very interesting patterns. And the other interesting I found there is that when you do that, you see that these patterns, that there are bacteria in there that do not adhere to these patterns. And they are probably, there's a good chance that these are mislabeled. So you say, well, I see that. So I have a graph on that here at the end, which I discovered on Wikipedia, and I thought that's nice. But turns out I had made that graph myself 2 years ago. Yeah, here. So here, this is a good example of your question. So here I made a graph for the firmisuits, and this is I'll get later to what this is, but you can see that most of these every pixel is a bacterium species. And you see that almost all of them are distributed along that line. So they show firmisuit behavior. But then you also see a few pixels that are like, no, I'm doing my own thing. And it's very interesting to look at that. Is this actually a different family member, or was this a mislabeled thing? And so yeah, you can go wild with that. And I've even made it easy because this terrible taxonomy database that is so hard to parse, I put it in CSV for you. And you can go wild. And this is like relatively virgin territory, because it's very difficult to discover something really new in biology if people have been doing that all the time already. But if you go do new things that no one has ever done before, chances are you could discover something. And that could be something as simple as, we thought this bacteria was in this family, but it should be in that family. But that's already a nice discovery. It's like you can send a pull request to these people. And Yeah, okay.
I have a question as well. You say like there's 46 possibilities with the 4 letters. There's only 21 being used.
Yeah.
You say there's margin for improvement. Do you mean evolution or is it something else you mean?
Oh, I must either I misspoke. However, I'm still going to tell interesting stuff on this. So you have this tremendous table, the codon table that we showed before, and I think really this is. I'm not a religious person, but the idea that all of life has been working according to this table for the past like 2 billion years is. Wow. I mean, it's a Rosetta Stone that people found one day. There was an interesting development in language, but this is nature's Rosetta Stone. Now, people sometimes look at this table, and if you look at it long enough, people develop mystic feelings for it. For example, people have found that you have these codons, which are 3 letters, and they have found that the second letter almost entirely dictates the acidity of the amino acid, if it will be a base one or an acidic one. And they look into this as sort of evidence for was this designed? Because if you see this table, it looks remarkably human. It doesn't really look like the fuzzy kind of nature thing. This is a digital table. It is like this, and it is in no way else. Now, lots of people have been doing math on it. For example, we know within the DNA that when copying mistakes are made, we know the frequency at which kind of mistakes are made. So we know that, for example, an A converts into a T far more often than a C into a G. So I'm making this up as I go along, but we know these statistics. And people have found that if you look at the most common spontaneous mutations, nothing happens. Because you mutate into the same amino acid. So that means even if a typical mistake happens, then maybe you move from one way of spelling leucine to one of the 5 other ways of spelling leucine. So then you made a sort of typo that didn't matter. And so there are people that say that this table is perfect. Because if you run simulations, if you assign this table at random, so let's say you just redesign it. It turns out that it's almost always worse in terms of the impact of mutations. So I don't think there is, I wouldn't say there is much room, maybe it could be improved, but not by me. There's maybe one thing I mentioned. Actually, on the previous chart that lists all the amino acids, there are actually 21 and not 20. So there is one special amino acid, and it's called selenocysteine because it has sulfur in there. And the way nature accesses that is that there is a magic DNA code which says, okay, the next time I put in TGA, that is normally the end of the protein. But in this case, when I have given you this escape sequence, next time it means selenocysteine, which we as computer people recognize as an escape sequence. We say, okay, if I put a backslash first and then an X, then you should interpret the following 3 things differently. And nature does this as well. And it is a rather sort of extreme attempt to cram in one more amino acid. But sometimes in life you can also see that, you didn't really think this through. But it's a very important amino acid, and they should have put it day. There is no day. But if there was a day, they should have just put it in the leucine one, because why do we have 6 ways of prescribing leucine? So maybe they could have made one of them selenocysteine, but there is no them. And this is where you see a gross hack, like an escape sequence, which in our world is also a gross hack. Yeah. Okay. Let me rant on that for a bit then. Sometimes when you look at software, you can see, okay, this is really version 3, because I see traces of version 2 in here. So, for example, you first had a single protocol API, and then later someone added IPv6 to it, and you can see that was a bit of a, well, they stuffed it in. If you now look at this 3-letter codon table, you can sort of imagine that leucine, again, does not need the 3rd character. Because if it starts with CT, it is always leucine. So you can see some traces in here that maybe in the very deep past we did not have 3-letter codons, but 2-letter codons. Maybe that was enough at some point. And if you look at this, people find that the cheaper amino acids, cheaper to make, all of them only need 2 letters of codon. And that is interpreted as a strong hint that maybe like 4 billion years ago, this table was just 4 bits instead of 6 bits. So like we did the painful transition from 16 bits to 32 bits to 64 bits. There you can look at this and convince yourself, well, life also went from this 2-position codon to 3-position codon change one day. Maybe. But it's a bit of archaeology. It's like digging up stuff in Egypt and trying to find out what it was. But if you look at, also do not look too long at this table. Because there have been people, as a little bit of a warning, you can see certain patterns in DNA, and if you look at that long enough, you start maybe to do mathematics on it. And there's people who invented a thing called the Z curve or the Z curve, depending on if you're from the UK or not. And there they say, well, if it's an A, you go up, and if it is a C, you go to the right, and if it's a G, you go like that. And when you apply those rules, you get indeed a sort of 3D whole rotating thing in DNA. And as far as I know, it means nothing. But you can get these feelings from it. That you look, hey, I'm learning something from DNA. I'm looking at the ancient 2 billion year old code. So don't look at it too long, would be my message. Yeah.
Go ahead. We'll figure it out.
So yesterday you also shown self-defense mechanism.
Yeah.
Right? And it had these marked areas.
Yeah.
Can you pull it up, please?
Yeah, so I can... Yeah, this one.
It appeared to me that all of those are of same length.
Yeah, except one.
Except one. Oh.
Yeah.
So is there some reason behind it, or how does it know? Okay, it starts here and it ends there.
Yeah, that's a very good question. And first, I'm gonna. So this is actually a remarkably digital piece of nature, where we know that the signature for a given bacterial species, the signature is always 17 or 18 bytes. 17 or 18 nucleotides. But it's not always 17. Which in our world we would say you get a struct and a fixed header and then 17 bytes of signature, and then or you would have to have some kind of termination mechanism. And nature is a bit fuzzier than that. So yeah, indeed the length is always the same, and the spacer, which the motive, which is the inverted bit, is always almost the same. Let me compare this to JSON. In a bad way. If you were like an old school C programmer, you would say, my array of signatures is just an array of signatures, and they are all this long. And you could also, but that is not what nature did. That inverted thing is not just a marker that says this is where this is where it begins. So it's not. It doesn't say begin. That marker is actually active DNA that empowers the signature. So the signature and the preceding bit of DNA together form a molecule that does stuff. So much like JSON repeats itself for every record and says, yeah, this is name, colon, name. This array of signatures is actually a bit of active code, signature, and then again a bit of active code, and then the signature. So it's the active code, which is here in green, inverted, is repeated again and again and again. So we would say make a for loop, or whatever, and it appears that in this case that is beyond nature. Nature just has to say here active code, active code, active code. But it's interesting to see that the lengths are indeed almost the same, except one of them. And so you can see that because the highlighted bit, the inverted bit, jumps by 3 characters on the next line. And for one case, it jumps by 2 characters, where you see that the signature is slightly smaller. And yeah, they get away with that. This is not a buffer overflow. I hope that sort of answers your question.
So highly speculative. If you say all DNA on Earth is exactly the same.
Exactly. You cannot in any way. Okay. Sorry.
But let me do the full question.
Yeah.
Like, could it be that we only got a portion, that somewhere in space the DNA got separated, and we only got like half a portion that spawned everything on Earth?
Yeah. So this is actually an area that biologists are a bit worried about. And I'll show you why. That's at the very beginning of the presentation. So, but they will not admit this in public because I'm not a real biologist. I can just leak the secret. And so many slides. Phew. Yeah, this is the good one. So, this is the current theory of how we think life started on Earth. It's one of these smoky vents on the ocean floor. And that's the current best theory that we have. And it is a rather good theory because there's energy, because hot water comes into the cold ocean. So there's a temperature differential. The chemicals are nice. And Darwin famously thought that life had originated in some small warm pond somewhere. That was his idea. This is actually a far better idea, and it seems likely. But the timeline is worrying. So we know that 4.5 billion years ago, the Earth was a far more hellish place to live than today, in the sense that meteors kept falling out of the sky, and the oceans were boiling. So 4.5 billion years ago, life was not, and even 4.2 billion years ago, the world was not a very hospitable place for life. Yet, in almost no time, did life start. So it says, in 300 million years. In 200 million years between the arrival of water, we had life. That's a relatively brief period. So people are very seriously studying, could parts of the DNA have fallen from the sky as inspiration? Maybe that could have. And we know that various components do indeed fall from the sky. So we know they are in meteorites and they get to Earth. If you ask any biologist, do you think that it helped? Then they will all say yes. It's likely that it helped. But if you ask them, how do you propose that all this evolved in only 200 million years from scratch? Then people go like, can we talk about something else? So this is, of course, the official line. So everything is by evolution, and we know how it works. But there is a little bit of worry. And the interesting thing is, of course, that since the DNA is still exactly the same after 4 billion years, because we could have seen it if it was different along the way. And people are like, apparently it's perfect. How did it get to be perfect in 200 million years? Well, so I'm sort of agnostic on it. I'm like, I don't know. I still find it a very exciting information storage technology. But your question was, of course, maybe we only got part of it. Maybe that's true. Maybe we put it on a diet even. Maybe there has been a simplification phase. I don't know. But it's still the case that if you take a piece of DNA from a bacterium and a piece of DNA from us, the only way you will be able to see a difference on it is that we actually have some ways of putting metadata on DNA. You can paint it. And that is used for all kinds of purposes. And bacteria and human beings, you do this painting in a slightly different way. So the metadata is somewhat different. However, lots of DNA has no metadata, and in that case, it's sort of exactly identical again. Yeah. Oh, yeah.
Hi, so what are your views on using CRISPR to change DNA in living beings bigger than a bacterium?
Yeah, so with CRISPR, you can, and base editors and prime editors, we are now gaining the possibility of altering DNA in living cells. But so far, just purely, not an ethical thing, just on a technical level, it works quite often, which is not very, you should be worried. If someone says, I'm going to CRISPR you would be like, well, what's the success rate? Well, 90%, and the other 10% just ruins some random bit of DNA. That's a bit of a lottery right now. We can still use it because in many cases we can take cells from your blood, take it out of you, then do the treatment in the lab on your blood, see if it worked, and if it worked, we put it back into you. And in that case, the 90% success rate is okay. because we can tell in the lab that we got a good one. But that only works, of course, for diseases that we can cure with cells in your blood. Let's say the problem is in your liver. We cannot take out your liver and then prepare it in the lab with a nice Chianti and then put it back and see if it worked. So my first thing would be the technology is not very precise right now. So that's the first thing, and the second thing is our knowledge of actual DNA is piss poor. So to give one interesting example, we know there is a gene that gives you blue eyes, or a mutation, and it's very reliable. If you have that part of the DNA and it is one letter change, then you get blue eyes. So you might think that this change is in the eye gene or whatever, or in the eye color gene. It is not. The change that is responsible for that is like 3 genes away in a completely unrelated gene. And why does that happen? If you make that change over here, then the DNA folds up somewhat differently from what we previously described, and then the gene that does not give you blue eyes does not get expressed. So this is a bit like if you have a machine and it doesn't work, and you repair it by changing a completely unrelated transistor. And suddenly it works again. If that is your level of understanding of the thing you are working on, you had better be very careful. Now, there is a small list of genetic diseases where we just really know it's just this one letter. We know that this is the thing. And my wife has one of these diseases. We know exactly which letter you want to change. And so you would maybe want to do that. But there is still the worry that your CRISPR thing makes more changes, for example. The first thing you're going to see where this is starting to work is, let's say you want to change a gene in a living person, you first have to get the DNA into the right place to make that happen. It's very tricky to do that, because you can inject DNA into one cell, but we do not want to fix one cell. We want to fix all your eye cells. Almost impossible to do right now, except for liver disease. And why does that work? When your body tries to get rid of something, it brings it to the liver. That means that we can actually rather easily do treatments of liver things, because you can inject them anywhere. And the body will say, I don't know what it is, I'm going to bring it to your liver, and there it might do the good thing. So I'm sort of enthusiastic about using these CRISPR and base editors and prime editors for a very limited number of diseases where we know that this single thing is it. But there is, of course, are lots of people that want to edit their DNA so that they become stronger or smarter or healthier or get bigger organs. I don't know, and that is probably not something we should be doing right now, simply because we don't know. So each gene does like 5 things. So let's say you would enhance the gene that gives you a very big ears. Maybe there are other organs that people want to make bigger. I don't know. But you change the DNA that gives you very big ears, and it might also make you very stupid, simply because we don't know. So that's the very long answer to your simple question.
We have about 10, 11 minutes left, so just you know where we are.
Yeah.
In your last talk, you were talking about n-grams and some other data analysis techniques that reminded me of computational linguistics. And I just came in, so I'm sorry if someone already brought this up, but I was wondering if anyone has tried using transformer models, AI, on DNA sequences.
Well, yes, so I have. And it was ridiculously successful. And by this, I do not mean you take DNA and you put it in ChatGPT and ask us, what does this gene do? Because we're not there. So let me tell you about a really fun problem. Yesterday, I showed that in bacteria, we have these very clean markers that say the gene starts here on various protocol levels, and at the end it says, and the gene ends here. And you can even with a terribly written C program, you can find genes in bacteria. We have no way of doing that in human DNA. We cannot by eye see that this is the start of a gene. It's extremely well hidden. There are some patterns, but in general, we as human beings cannot recognize where a human gene begins, which is ridiculous. This is one of these other biology secrets that they don't talk about. You can read about this on Wikipedia. It's called gene discovery or gene finding. And so I did a simple experiment. I took the known places where we know that genes start. And I trained a very simple recurring neural network on half of that data. Say, can you reliably say this is where a gene starts and this is where it didn't start? And in 2 days' time, I had something that was like 70% accurate, which is ridiculously good. I could not believe it, and maybe I still don't believe it. But it looks like recurring neural networks, of which transformers are the bigger version, but there are smaller ones. But even I could use one called GRU, GRU, which is also a Russian intelligence agency, but it also finds genes apparently. So yeah, this could be very successful even with simple models. And the hope there, of course, is that you take a simple model that learns this thing to spot genes, and that the model is simple enough that we can find out what it learned. Because it's nice if we have a network that can find genes for us, but even nicer if it tells us how it does that. So the message really is also to you, you can actually do this, and it's simple to do, and you could actually have results there, because many of the people working on AI are a bit mental, and they are trying to get ChatGPT to do this for them, but if you use like an actual neural network, you can have a lot of fun there.
Can you not take the blood out of the person, do the CRISPR treatment on the blood, and then process the DNA and read it? And then, as you said after the talk yesterday, you can read the DNA and only look for the bits you know should be in there that you inserted, and then look in which bit they ended up in.
That's what they do. So that's also why these treatments are ridiculously expensive. Because you do the treatment in the lab, you fix a certain antibody or you make a new one, and then you sequence the whole thing again to see if you got it right. And the success rate of this has been like pretty good, in the sense that we do this now for lethal disease. Because we do this for where you think, you're going to die anyhow. So it's worth it. But there have also been cases, sadly, where this screening was not perfect. And then you end up with people actually dying from your treatment. Yeah.
Could you insert something like a plasmid inside DNA so that it would be separate?
Yes. So the question is, and this is actually this is a real fun question. So sometimes we have a broken gene in a person, and we would like to fix that broken gene. One way, of course, is to really edit the source code. Say we're going to fix the gene that is there. Which is difficult. Another way of doing it is installing an extra gene. And to everyone's sort of surprise, there is a virus called adeno-associated virus. So this is a virus that somehow lives together with another virus. Nature can finds a way somehow. And the adenovirus is a normal cold virus, and it turns out that this adenovirus actually, as one of its actions, it has this associated virus which delivers a mini circular chromosome into a cell. And everyone is super excited about this, because if in that little circular genome you put a fixed gene, so you know a person has a broken gene. Well, we cannot maybe fix the broken gene, but we can give you a fixed gene. See if it works. And this is sometimes successful, sometimes not, but here's where it gets like really interesting. This little circle of DNA that we can insert that way is like really tiny. And it's so tiny that it's actually too small for most genes to fit in. And that has led to like huge efforts to give you a shorter fixed gene. To see, can we take the gene and it's broken, can we make it shorter so that it still works? And then insert that, which is a neat hack, which sometimes works. And the other one is that you infect someone twice with 2 of these different viruses. You get 2 of these small rings that get in there, and you coat them such that the DNA attract each other and that they plop together form a bigger circle. This story would have been more fun if it worked. But everyone is like, it should work. And so this is specifically for some eye diseases, because there are a lot of them, and where we can very often point to a single gene as the fault, and that we would like to deliver that single gene with this virus. And because it's a virus, it infects all of your eye, and could deliver that extra gene to lots of different cells in hopes that enough cells get the fixed gene. So yeah, we do try that with this virus, but it turns out that our body really hates viruses. And it's like very good also at stopping them. So it is a hit and miss. But yeah, this is, and everyone, there's a huge field now in adeno-associated virus engineering, because people want to make this virus bigger so that they can increase, and they actually call this the payload. So yeah, the people are working on that. Yeah.
So yesterday you said we should get involved, we as hackers, computer people. How would we do that concretely?
Very concretely, if you would, let's say, there are I think 2 ways of doing it. One of them is learning the extremely terrible set of bioinformatic tools that are out there. One of them is a whole community called Bioconda, and there you can get version hell and everything. And you can find courses on that, and this is sort of and if you do this, you could quite easily turn it into a career. Because once you are good at that stuff, you can work at big pharma, and because you work with the known tooling. This may be for some, there are lots of YouTube movies, so this is sort of but you end up with this crappy software. But it's the crappy software that everyone uses, so it is still worth it. I went the other way around because I'm damaged like that, so I wrote all my own software. But I with that, I convert a lot of the crap data formats to CSV files. Which means that if you go to skewdb.org and pork the repository and stuff, and sadly you also have to ask me a few questions because I need to give you a few pointers, but then you can get going the moment you understand CSV files. And the one thing that I would love to do, but I maybe not have time for that.
Up the QR code. You want to put up the QR code?
That's a very good one. So before I do that, again, this is the spiral form of a bacterial piece of DNA. The first thing I would do if I suddenly had nothing else to do, I would write code that would extract the DNA from the white spaces here. So that they're not genes, we don't know what it is. And just collect that from first start with one bacterium, do statistics on it, collect it from another bacterium, also do statistics on it, maybe start comparing strings. Are there patterns in there? Are there engrams that happen a lot? In the world of biology, they don't call them engrams, they call them k-mers, because they have their own words for things. But that's what the first thing I would do. I would now look at the white spaces and see where else do they show up. So I put up the QR code because I think we're sort of heading towards the end. Yeah. Oh, there's
2 minutes.
Yeah. So this is the from. If you have this QR code from there, you can find the other things. And I hope that gives you some inspiration. And also feel free to email me if you want to get going. Let me know. I can give you pointers because it's not the most accessible field ever. So if you want to find the right data, you have to find the right FTP server, even though it no longer does FTP. And if you go to one of the most frustrating things, so let me warn you about this one. There is the big genome server of the US government, the NCBI. You click on the link, genomes, and then you go to bacteria all, and you click on it, nothing happens. And it turns out you have to wait like 5 minutes. Because it loads 100,000 file names, and apparently that's a lot of work for that server. But at first, for many times, I thought the server is down, it doesn't work, and then later you discover a tab with all these bacteria in there. The rsync interface works a lot better. So getting started takes a little bit of time, but then the data is there, and if you can grep, and if you can awk, then you can do some amazing things. And I think with that, I'll round it off. And thank you so much for listening to this stuff for an hour. It makes me very happy.